Let's look at the blood vascular system. So the blood vascular system, what we're talking about is, of course, blood. We're talking about the heart, which acts as the pump, the overall circulatory system, and then the lymphatic system, which works within this circulatory system. Blood is a connective tissue, and it's many cells suspended in a plasma matrix. Functions are transport and regulation. Transport, we're talking about transport of gases in solution, which could be exchange of oxygen and carbon dioxide. Nutrients, we transport nutrients from the digestive tract into systemic circulation. Waste products, blood collects wastes and sends them to the kidney and the liver for breakdown and disposal. And of course, hormones and enzymes we just learned about with the endocrine system. Blood transports these hormones that are produced by various glands to their target tissues. Regulation, we're often talking about volume and body fluid. So maintenance of homeostasis, which maintains the osmotic balance through plasma proteins. So it maintains a certain level of fluid within the vessels of the vascular system. Temperature is regulated, and it's maintained through peripheral vasoconstriction, so constriction of some of those smaller blood vessels. And it's lost through peripheral vasodilation. So the temperature can actually be lost when some of those blood vessels dilate and they release some heat from the body. Acid-base balance, the exchange of hydrogen ions to level out the acidity of the blood. Defense, white blood cells, of course, all part of the body's immune system. Antibodies and antigens are carried to the site of action. And clotting, platelets prevent extreme loss of blood. Edema is a term that we should bring up because when it comes to maintaining homeostasis and maintaining the appropriate level of fluid within the body, an abnormal accumulation of fluid in the cavities and intercellular spaces of the body is called edema. So we have different types of edema. If edema is occurring in the the peritoneal cavity, so the abdominal cavity, then we often call it ascites. But edema could be all throughout the body. It could be in the legs, it could be in the trunk, it could be in the thorax, etc. So that's an abnormal distribution or accumulation of fluid in the cavities and intercellular spaces. So this is from your textbook. This is the blood components. So if we look at each, we've got blood at the top, and then we break it down into formed elements, so little cells that are suspended in the plasma. When we look at plasma, it's made up of serum and fibrinogen. If we look up the formed elements, we've got leukocytes, which are white blood cells, erythrocytes, which are red blood cells, and platelets. And then from leukocytes, we have granular leukocytes, aka granulocytes, and then we have agranular leukocytes, also known as agranulocytes. And we'll go through each component of those. So plasma, that matrix that suspends the cells of the blood, is 90% water. And within it, you'll find carbon dioxide, amino acids, glucose and fatty acids, waste materials, hormones, enzymes, antibodies, and antigens. Plasma proteins, the most common plasma protein is albumin, and it maintains osmotic concentration of blood, and it's produced in the liver. So albumin is a blood protein that encourages water to come near it or get attracted to it. So it encourages water to maintain itself in the blood vessel specifically. If we lose albumin, then we're going to lose blood volume in the vessels. Fibrinogen and prothrombin are involved in clotting the blood, and they're also produced in the liver. And immunoglobulins are antibodies produced by the immune system produced in certain white blood cells. So this is whole blood. When we take it from the animal, if we spin it down using centrifugal force, then we're going to get a breakdown of the matrix or plasma, which is roughly 55% of the total blood volume. We get a buffy coat, which is a very thin layer of leukocytes and platelets. And then we get this big chunk at the bottom, which are red blood cells, which is roughly 45% of total blood. So blood cells, we've got erythrocytes, which are red blood cells, leukocytes, which are white blood cells, thrombocytes, which we refer to as platelets, which are actually fragments of larger cells. Erythrocytes are biconcave, so they have this disc-like appearance, and they're pigmented by hemoglobin. That's the protein that carries iron. Mammals, we don't have a nucleus in our red blood cells, but reptiles and birds have a nucleus. And the RBCs carry oxygen and small amounts of carbon dioxide. Erythropoiesis, or the creation of red blood cells, occurs in the bone marrow, and the RBCs start off as nucleated cells, and then as they grow older, they lose their nucleus. The lifespan of the average erythrocyte is roughly 120 days in the mammalian species, and then they're destructed. They're destroyed. Typically in the spleen, certain organs will kind of destroy them and then reuse their parts. Production of red blood cells is controlled by erythropoietin, which is produced in the kidneys. Kidney failure, animals that have short-term acute kidney failure or long-term sort of chronic kidney failure, over time they will become anemic. So they will have less red blood cells produced because when their kidneys are not functioning, then they're not producing enough erythropoietin to carry on the development. Oh, and I'll just point out here, so all of these cells start from a myeloid stem cell in the bone marrow, and then of course they get shifted into various cells. So here we've got pronormoblast, basophilic normoblast, polychromatic normoblast, also known as rubricites, and then of course down to a polychromatic or multicolored red blood cell, and then to its regular smaller red blood cell format. Leukocytes are white blood cells, and within this category we have granulocytes and agranulocytes. Leukopenia is a small number of white blood cells present in blood. So that term leukopenia means that they are lacking in white blood cells. Granulocytes are produced in the bone marrow. 70% of all white blood cells are granulocytes. And examples of our three types of granulocytes are neutrophils, which make up 90% of all granulocytes. They're able to move into tissue to phagocytize foreign material. Eosinophils are involved in the regulation of allergic and inflammatory reactions. They secrete antihistamine, and they also have a role in controlling parasitic infections. And then basophils also secrete histamine and heparin. So just looking at them here, we've got typical appearance of a mammalian neutrophil, typical eosinophil, which has these big, nice red components within the cytoplasm, and then a basophil has a slightly purplish tinge throughout. Agranulocytes, the two examples are lymphocytes, which are formed in lymphoid tissue, and they originate from stem cells in the bone marrow. They're involved heavily in immune reactions or immune response. T lymphocytes create cellular immune responses, and B lymphocytes are involved in the production of antibodies. Monocytes, on the other hand, they are the largest leukocyte. They're phagocytic, which means that they actually will engulf and destroy various microinvaders. And monocytes are able to migrate to tissue to help with this phagocytic effect and help control the numbers of foreign invaders in the body. Lymphoma is a condition I'm sure you've heard of before. So this is an abnormal and uncontrolled growth of T and B lymphocytes in lymphoid tissue. It often results in the formation of a tumor or swelling of the lymph nodes around the body. So lymphoma is a huge complex topic. There are so many different forms of lymphoma, different types of lymphoma. So that's something that's definitely worth investigation if you have an interest in that. Otherwise, moving on, thrombocytes are also known as platelets, and they are fragments of these really big cells called megakaryocytes. So you can see here, this is the megakaryocyte. It's this huge multinucleated cell that circulates in the body, and it releases these small little fragments around the outside of its cytoplasm. And you can see the little fragments there. They end up becoming platelets. So very tiny little pieces of megakaryocytes are platelets. Platelets themselves, they're not cells, we have to remember, and they also have no nucleus. They are involved in blood clotting, and the term thrombocytopenia refers to low numbers of platelets. So those are the basic components of blood. It's good to know what each blood cell is called and definitely what each blood cell is involved in within the body. And of course, the components of blood overall. So now we're going to move on to the main attraction, the heart. So the heart is contained in the thoracic or pleural cavity. The apex of the heart forms a loose attachment to the diaphragm, and it lies slightly left to the midline of the animal. So remember that the midline is kind of that line we draw from cranial to caudal, right along their umbilicus or their belly button. So that's the midline there. So the heart actually lies slightly left to the midline. And then the apex, which is the pointed aspect, the caudal-most pointed aspect of the heart, forms a loose attachment to the diaphragm. The heart overall is protected by a fibrous connective tissue called the pericardium. So this little diagram here is able to show just overall that the heart has this thin membrane, fairly thick actually, but a protective fibrous membrane that covers the heart and the start of the vessels. So this pericardium, this fibrous membrane that protects the heart, is 3 layers and a fluid layer. It offers protection, lubrication, and movement of the heart, and it actually prevents friction of the heart when it rubs up against other thoracic tissue. So if we look closely here, we've got the pericardium itself. We have a little pocket of pericardial fluid. If there's too much fluid accumulation, that's called pericardial effusion, and that's typically related to a disease process. So here's a normal x-ray of the thorax. So we've got the black, remember on x-ray, air turns out as black. So we've got black in the lungs. We have black in this tube here, that's the trachea, so also known as the windpipe in common terms. And then this big guy here is the heart. So with the heart, it should look slightly oval with a pointed aspect that's pointing ventrally and caudal. This is an image of a dog with pericardial effusion. So that thin little layer of fluid that sits within that fibrous protective layer around the heart has accumulated too much fluid, and it ends up creating this huge ball-like appearance of the heart. So it gets really round and really quite enlarged. So that, as you can imagine, is a significant disease process. And then when ends up happening, because the pericardium can't stretch too much, it will just start to accommodate the fluid and compress the inside of the heart, which makes pumping the heart very ineffective. So looking at I'll just go back to that pericardium. So this is the outermost layer, the fibrous pericardium, and then we have a middle layer, which is the parietal pericardium. It's within this pericardial cavity that we have some fluid, and then right attached to the heart, we have the visceral pericardium. Next up, we have the thickest layer of the heart, which is called the myocardium. And myo, we know, refers to muscle. So myocardium is the muscle layer of the heart. And then the innermost layer of the heart, the area that actually touches the blood as it passes through the chambers of the heart, that smooth layer is called the endocardium. So looking at the components of the heart, we have the left and right atria, which if we're talking about singular form, that's the left atrium and right atrium. We have the tricuspid valve. Right here, we have the left and right ventricles. We have the bicuspid, also known as the mitral valve. And then we have the pulmonary valve and the aortic valve. Attached to the heart, as part of the working pump of the heart, are the pulmonary veins and the pulmonary arteries, as well as the cranial and caudal vena cava. So looking at the flow of blood through the heart, the vena cava takes deoxygenated blood, from the tissues of the body, and it collects it. So the vena cava runs all the way up from the tail. It runs along the dorsal aspect of the abdominal cavity, just past the abdominal cavity. And it brings all that deoxygenated blood up from surrounding tissue and empties it into the right atrium. So we've got the body tissues giving their deoxygenated blood to the vena cava, emptied into the right atrium. From there, the right atrium over here contracts, and it pushes that blood through the tricuspid valve and into the right ventricle. From there, the right ventricle contracts, and it pushes blood up through the pulmonary valve into the pulmonary arteries. Now, from the pulmonary arteries, the blood goes directly to the lungs to acquire oxygen. So as of this point, the right side of the heart is all deoxygenated blood, and then the blood goes to the lungs through the pulmonary arteries to acquire oxygen, and now it's going to enter back into the heart as oxygenated blood. So the pulmonary veins from the left and right side of the lungs are going to bring blood into the left atrium, and you'll notice now that this is red, so it's oxygenated. The left atrium will contract, and it will push blood through that bicuspid or mitral valve into the left ventricle. And then the left ventricle will contract. It will push blood up through the aortic valve, and now this oxygenated blood will get distributed all through the body as it works its way through the aorta. So the aorta is the largest artery in the body. The vena cava is the largest vein in the body. And both of those run all the way down the length of the body and all the way up the length of the body to ensure that deoxygenated blood is taken back to the heart for reoxygenation, and the oxygenated blood is circulated around the body to the tissues. The thickest area of the heart is the left ventricle. So that's the thickest layer of myocardium, because if we think about it, when the left ventricle contracts, it has to create enough force to ensure that that blood is sent through the aorta and all the way down, pretty much all the way down to the tail, to ensure that oxygenated blood gets all the way to the tissues in the body. Whereas the right ventricle, when it contracts, it just has to send blood to the lungs. Okay, and it's very close to the lungs. Looking at the valves, so the valves all have different names. We have 4 valves, and we have 2 types of valves. So we have the atrioventricular valves, which I'll go back here, and they sit between the ventricles and the atrium. So atrioventricular valve. And then we have the semilunar valve, which sit between the ventricles and the vessels. So the different valves that we have are the tricuspid valve, which is named because it has 3 little flaps. So 3 little cusps, tricuspid, and that's what controls blood flow. And then the bicuspid has 2 flaps. Okay? And then the others, the aortic valve and the pulmonary valve, are also known as semilunar valves. A heart murmur is a sound that results from turbulent blood flow in the heart, and it's often associated with a poorly closing valve. So if any of these valves don't connect in full, they're 3 little flaps, they're 3 little pieces of tissue that close to create a beautiful valve. If they don't close all the way, then small amounts of blood will continuously get out of those valves, and that's what causes a heart murmur, which is a swoosh, swoosh sound. Okay, so let's look at conduction of energy through the heart. So the heart relies on energy and nerve impulses to stimulate the pacemaker of the heart. So the pacemaker is also known as the sinoatrial node, and it's in the right atrium of the heart. So it's embedded in the myocardium of the right atrium of the heart. The fastest heart cells to contract, they set the beat for the heart. So those cells of the myocardium that are within the sinoatrial node are the very fastest cells to contract when that electrical impulse is sent to the heart. So normal cardiac function, the impulse is sent to the sinoatrial node, and then it works its way down to the atrioventricular node, and then that electrical impulse crosses through the bundle branches, through the bundle of His, into the bundle branches on either side, and then into the small little Purkinje fibers. So throughout all of this, that electrical signal is getting sent from the right atrium. It's going through the two atria, and then it makes its way crossing the atrial borders down into the two ventricles. And as the electrical impulse gets sent through those areas, it's causing those areas to contract. The way we measure this in vet medicine is to look at an electrocardiogram, or an ECG, and it gives us an idea of how the energy is being processed through the heart. So we can look at this particular video in class. Okay, some diseases of the heart. We have hypertrophic cardiomyopathy, which is the thickening of the myocardium. So that muscle layer is very, very thickened, and it results in smaller ventricular chambers. So the ventricles now have less space in the middle of the ventricle for blood because the muscle layer is too thick. So it results in inefficient pumping of blood to the surrounding tissues. So this is a normal myocardium of the left ventricle, and this is hypertrophic cardiomyopathy, whereby the heart muscle in that ventricle is thickened. So you can see now there's just this tiny amount of space for blood. Dilated cardiomyopathy results in a thin and floppy myocardium. So in this case, there's tons of room for blood, but the heart is actually unable to contract fully and efficiently. So again, we've got a normal ventricular space, normal myocardium, and then we have dilated cardiomyopathy, where the myocardium gets thinned out and floppy, and those valves can no longer close effectively, and the heart can no longer contract effectively. So we also have to measure the body's blood pressure, and the blood pressure, we're looking at the arteries, so the pressure in the arteries during contraction and during rest. So typically we're talking about diastolic and systolic, or systolic and diastolic. In a normal dog, let's say, like a normal big dog, it would be normal to have 120/80. And in this case, the first number is always systolic and the second number is always diastolic. Systole, which results in systolic pressure, is the contraction of the ventricles. So it's blood getting pushed out into the body. Diastole, or diastolic pressure, is the pressure during relaxation of the ventricles, where the ventricles are filling up with blood. So looking at our various types of blood vessels, arteries carry blood away from the heart. So if you remember, the pulmonary arteries took blood from the right ventricle and sent it to the lungs. And veins always carry blood back to the heart. So we've got the vena cava, of course, which is the biggest vein in the body, bringing blood up to the heart. And then you've got the pulmonary veins as well, that of course bring oxygenated into the right or the left atrium. Capillaries are involved in cellular exchange, and they are one cell layer thick. So they work based on the principle of diffusion. Arteries are thick vessels. They're the thickest vessels in the body, and they contain a muscle layer. They're very flexible to adjust to changing blood pressure, and they branch into arterioles. So as an artery gets smaller, you can see you've got the aorta, which is the largest, and then it goes into a large artery, and then a small artery, and then it branches into an arteriole, and then it becomes a capillary. Gas exchange takes place, nutrient exchange takes place, and then it connects back up with the vein system. So in arteries, this is where we can feel the pulse in an animal, is within an artery. Veins carry blood to the heart. They're under low pressure. They have valves in them that are there to direct blood flow, so they don't rely on a muscle layer within the vein itself. They rely on valves to ensure blood flows in one direction, and they rely on skeletal muscle in order to pump the blood throughout the body. So when your leg falls asleep, because you've had it crossed for too long, This is your body telling you that you need to get up and move because your veins, which return blood to the heart, are now becoming stagnant because they need contractions of your skeletal muscle to help push the blood back up to your heart. Veins do not have a pulse, so we cannot feel the pulse in a vein. Alright, so then vascular anatomy overall, the main trunk of the aorta will arch dorsally and then it travels caudally. So it comes up from the heart, works its way cranially and dorsally, and then it sort of flips its way back behind the heart and works its way all the way caudal throughout the body. It has numerous branches that emerge in the thoracic and lumbar areas of the body. The main trunk of the aorta splits at the hind limbs to create the iliac arteries. So you can see that here. The aorta runs all the way down the length of the body and it splits at the left and right leg to create the iliac artery. And then the coccygeal artery emerges at the caudal aspect of the aorta. So it becomes this tail artery called the coccygeal artery. Venipuncture, when we're talking about taking blood, we are 99.9% of the time talking about taking blood from a vein. And venipuncture is, of course, specifically to a vein. The cephalic vein is the cranial-medial aspect of the forelimb. The medial saphenous vein is the medial aspect of the hind limb. And the lateral saphenous is the lateral aspect of the hind limb. So we've got cephalic, and you can see this is the dog's elbow and her hand here. Here's the carpus. We've got this nice big cephalic vein that runs to a little bit proximal of the carpus and then splits. And then this one is a cat. It's hard to picture, but this is the cat's anus over here in its tail. And this is the cat's hock. And she or he is just holding the leg and taking blood. And it's on the medial aspect of the hind leg. And then the lateral saphenous we use on the dog, and that's on the outside or the lateral surface of the hind limb, just proximal to the hock. So here's the hock, there's the foot, and then of course there's that lateral saphenous vein. Good little memory tool, the saphenous is close to the penis. Cephalic is not. And then, of course, we have the jugular veins, which are on the ventral aspect of each side of the neck in the jugular groove. They are close to a big artery called the carotid artery. So, when we're taking blood from these locations, we always have to be careful to avoid accidental injection into the carotid artery. So you can see here on this guy, the trachea is here. And then when we press down in the thoracic inlet, which is identified by the manubrium here, and just you shift left or right, you can see that they've raised that jugular vein on the cat. And that is it for venipuncture and for circulation overall. So now we are going to shift into the lymphatic system. So the lymphatic system returns excess tissue fluid back into the circulating blood. Lymphatic fluid we call lymph. And lymph is very similar to blood plasma, but less proteins and more lymphocytes are present. The functions of the lymphatic system are to return excess fluid into blood circulation, remove bacteria and foreign invaders or foreign particles through lymph nodes, produce lymphocytes, and transport products of fat digestion and fat-soluble vitamins from the intestines. So here's a good idea of the very basic outlook of the lymphatic system. In our main thoracic duct that branches down, and then as the little ducts sort of differentiate, they're attached to lymph nodes as we go through. So the lymph nodes are little mechanical filters. And on the body we have various lymph nodes that are present. And some of them we feel regularly to help determine the health of the animal. So every lymph, lymph in general, works its way through lymph vessels throughout the body. And then as it's working its way through circulation through these lymphatic vessels, it circulates through a lymph node. So lymph always has to circulate through a lymph node. When it goes through the lymph node, then there are cells in there that will help phagocytize debris, and it essentially helps clean the lymph. So when an animal has an infection in a local area, oftentimes their lymph nodes will swell. That's like you guys, if you get a cold and you say my glands are all swollen, we're actually talking about your lymphatic nodes, your lymph nodes. So in dogs and cats, and horses too, we often check their lymph nodes for enlargement, and that gives us a bit more information about something that might be going on. So common locations, submandibular lymph node, it's right at the very caudal base of the mandible, and it's just below the mandible. Typically on cats, it's about the size of a pea. On dogs, it's about the size of a kidney bean. Prescapular is just in front of the shoulder or the scapular region. And same idea, same size. And then popliteals, I would say this isn't my favorite drawing of where popliteals are. They're a little bit more caudal, and they kind of sit within those hamstring muscles. Okay, so we can palpate those on normal, healthy animals and then compare them to animals who are sick or might have inflamed lymph nodes to identify changes. Then there's the spleen. So this is the largest of the lymphoid organs, and its functions are to store blood, and it destroys old red blood cells, and it removes foreign material from circulation. Now the spleen in dogs, cats as well, but I mean in general we don't really hear about the spleen much until something goes wrong with it. It's just this organ that kind of looks like a tongue, and it lives within the abdominal cavity. So normal spleen is roughly the same size. I mean, it depends on the animal, but it's pretty much the same size as that animal's tongue. Okay? The unfortunate thing is, especially in large breed dogs, a lot of them, the spleen is a common site for cancerous tumors. So what often happens is you hear about the spleen most often when something's wrong with the spleen and it looks enlarged on x-ray or ultrasound and inevitably needs to be surgically removed. So look at the size of that spleen. That's crazy. And you can just imagine a dog with a spleen that big, how crummy they must be feeling when they have something that big in their abdomen. The thymus is another lymphatic organ, and it's one that's particularly important in young animals. It aids in the production of T lymphocytes, which results in that cell-mediated immune response. So you can see it up here. It's just at the base of the trachea, right within that thoracic cavity. It's this very tiny little gland with a very big purpose in young animals. The tonsils are lymphoid tissue that are around the junction of the pharynx or the throat and the oral cavity. Oftentimes we see them if they're reddened or enlarged, just like with us. And the tonsils are the first line of defense against invaders in the mouth, and the goal is to prevent those invaders from entering into the respiratory tract and the digestive tract. This is what they look like in an animal up close. So we've got the tongue, and then of course tonsils on either side just before. And that is it.